The goal of this siRNA screening protocol is to investigate the impact of gene silencing on cancer cell line spheroid growth. Compared to traditional two-dimensional cell culture techniques, cancer spheroids display conditions more similar to those encountered in tumors in vivo.
This method is useful for the functional assessment of frequently altered genes in human cancers. And more importantly, it can be used to determine which genes are potential oncogenes or tumour suppressors under more complex culture conditions such as that seen in three-dimensional culture, and in particular under hypoxia.
The main advantage of this technique is that it's readily adaptable so that researchers can use it to study their particular genes of interest in their preferred cell lines. We believe that it builds on current targeted siRNA screening approaches.
It's generally appreciated that targeted screens, rather than whole genome screens, allow scientists to ask specific questions and generate more robust results. We have analysed the effect of gene silencing on spheroid viability and size, but you could also use biometric dyes for a different screen readout, such as apoptosis.
Demonstrating the procedure is Patty Way from our laboratory.
Begin by defrosting 96 well plates containing the siRNA library at room temperature. Then spin for 5 minutes at 1000 times G using a benchtop centrifuge. Use a multi-channel pipette to add 10 microliters of reduced serum medium containing the optimized transfection reagent to the wells of each plate. Incubate for 15 minutes to allow transfection complexes containing siRNA to form.
As with any screening pipeline, spheroid forming capacity of cell lines and transfection conditions should be robustly optimized prior to performing the full screen.